Hello again. So this time, indeed, I will change a bit from what we see in EEG to what we see in fMRI. So let's do like this. So overall, so this is the outlook of the talk, but I will just go through this. So overall, we see these macro scale functional networks detected in resting state functional magnetic resonance imaging, right? You all know fMRI. So basically the signals we detect with fMRI, they appear correlated. So if we have correlated fluctuations, we assume there is a functional connection. So a functional network is understood as a group of regions activating and deactivating together, right? So these have different names in the literature: resting state networks, intrinsic connectivity networks, co-activation patterns, even functional gradients are the same kind. I will even show there are different techniques to detect them with ICA and things, but overall there are these patterns. And these patterns have a functional relevance. So even for psychiatry, this is a paper in the Lancet Psychiatry, who proposed that these modes are associated to different aspects of brain function, such as salience, negative affect, attention, positive affect, even cognitive control. So this is also related to cognition, which is not just psychiatry, but also like related with cognitive decline, right? It may be important. And the thing is that these networks are consistently detected in fMRI studies. They appear disrupted in disease. They seem to be modulated by drugs, task, transcranial magnetic stimulation, by music even, as I will show. They are detected across mammals, so there's now some consistency also that what the patterns we detect in rats and in monkeys are similar or analogous to what we see in humans. Their origin, though, remains under debate. And there's still an ongoing question about what organizes activity at this scale, right? So, but for example, one thing that we rarely see, I guess, is to see really the fMRI signals in the brain, right? How they look like. So here I use the standard pre-processing from resting state fMRI, which is alignment to standard space, MNI space, motion correction, removal of artifacts, components, and I simply remove the mean of the signal. So every time we see something red means that the signal is above the mean, and in blue means that it went below the mean. And it looks very chaotic, right? It looks very noisy, but if we look at this obsessively for a long time, we see that there are some patterns that reappear. So here I put a pause here, a marker to see here a point where we see the default mode network. Here in the top plot, we see that there is an activation here in the precuneus, angular gyrus, and prefrontal cortex, here in the middle orbitofrontal cortex. It appears very noisy, right? In another subject here, just to highlight, for example, we see that a similar pattern appears. So there is some consistency in these patterns. And the thing is, like, it's nice to see. So these patterns are not always active. They seem to appear like from time to time. And sometimes they appear positively, and sometimes they appear, we see the negative of these patterns, and we see other patterns. Perhaps the DMN is clearer to see. So basically, when we capture and we do like we have here, I'm showing here the signal in one voxel, and here I show the signal in another voxel, and we can see that the correlation is not static. So the correlation overall is 55. So there is some correlation, but there are instances of time where they are really synchronized, and that's when, for example, here, when the default mode network would appear or not. So if we apply just a static functional connectivity measure, and we put indeed a seed in one of the nodes of the DMN, we will observe just in a single scan that there is already a pattern that resembles the default mode network. But the thing is that this static FC does not inform how stable this correlation is or how often this pattern actually occurs. So it's hard to quantify based only on these static measures. So when we look at this, like the slices that I was showing before horizontally, now I'm stacking them up together just to illustrate what is an MRI volume that we detect at each instant of time. So at each instant of time, sometimes this is quite long, right? We have different volumes. And as I show here, for example, here then I show just a single slice just to highlight, but there are different instants of time where we see a similar pattern. And then there's also something interesting, which I will develop on this a bit later, but that we see a negative representation of the default mode network as well, sometimes following the positive activation of this network. But it's important to be able to detect the time points when this network occur. So if we apply some technique to detect all these time points and we average across all the time points, we detect something that then really looks like in voxel space like the default mode network. This is one, but there are others. So it's important to characterize when these networks occurred, and what I was showing there is I used a method, developed a method, let's say, or gave it a name, which is we termed Leading Eigenvector Dynamics Analysis, or LEiDA, more briefly. That basically what it does is that we take the fMRI signals that are recorded in each brain area. So we parcellate the brain, again, like I explained before, we use a template, here again, 90 brain areas. And each signal, we apply the Hilbert transform, which allows to represent the signal at each instant of time as a phase and an amplitude. And then we compute at each instant of time the cosine of the phase difference between brain regions. So it's this matrix that we call instantaneous functional connectivity, which means if two areas are in phase, it's 1. If they are in antiphase, it's -1. And if they are with 90 degrees difference, it's 0. So it creates this matrix, which is quite noisy, as you see, like, but if you again look at it from time to time, like for a long time, we see that there are some patterns that reoccur. But then if we take, instead of considering this whole matrix, if we take only the leading eigenvector, so the eigenvector with largest eigenvalue, so matrices can be decomposed into eigenvectors, right? And we sum them together to reconstruct the matrix. And if we take just the largest one, the one with more magnitude, and we just here plot it over time, because this time is a vector, is a 1 times 90. So we can really plot it on the brain and analyze it better. It reveals like subsets of regions that are synchronizing together. And again, looking at it, we see from time to time the formation of patterns that resemble the somatomotor or the lateral frontoparietal or the default mode network or the visual. But again, it's super noisy, as you can see. So then if we apply a clustering algorithm, so we have all these patterns occurring over time across different scans, different subjects and that. So we have one for each time point. And then we put everything in a clustering algorithm to tell us, like, divide this into 7 networks. Just a second, because this is evolving too fast. And for example, the cluster centroids, which is for each cluster, there is we can do the average of all the patterns that were assigned to a given cluster. And this gives different network representations. So in general, usually the one with more occurrence, the one who appears more often, is just a network where everything is in phase. So here I'm plotting in red or links between the areas that shift in phase with respect to the whole brain. Because for some reason, this is what is revealing like known resting state networks. So here we cluster, and there is a global, there is a visual, the lateral frontoparietal, the default mode, a limbic network, somatomotor, and the dorsal attention network, right? And this is like it aligns with what people have been observed with other methodologies. So it's one more methodology. And this, in a way, allows to first approach brain activity as a trajectory in a state space, in the sense that if each of these networks is represented by one of these crosses, okay, and then we will start over time. So I'll take the leading eigenvector at the given instant of time and place it, right, at each instant of time closer to these different states. So we can see that over time, it seems that brain activity is a kind of transitioning between different patterns of activation. So this is a way to that is used to represent multi-stable dynamical systems and to see how they transition between distinct patterns of activity. But one of the advantages of this, so these trajectories can be represented over time here with shaded bars, where, for example, every time we have a blue shade, it means that the signals were closer to a pattern that resembles here the resting state network, either the positive or the negative representation, because the leading eigenvectors are non-sensitive to the sign of it. But this allows, so for each fMRI scan, which is nice, to calculate the state probability, the functional connectivity state probability, the duration, and also switching profiles. So this means that we can get a single scan matrix, let's say, so from the 1 GB of the fMRI scan, we reduce it, and we obtain these measures, which are analogous to the measures we can use from graph theory or from other things, but that quantify how often each of these networks occur during this specific scan. And this allows them to compare statistically between conditions, which is nice. So these different measures that we take, just to go a bit more into detail, and also here we try to link with the formalism, let's say, of dynamical systems theory, right? So the term that is used for the probability of occurrence is the occupancy of this state. And the occupancy is basically the proportion of time points that are assigned to a given state in each session, S, right? So it's like here counting how much, what's the percent of blue in the whole scan. And the dwell time, so to see how long when we enter that state, how long do we remain in that state, right? The dwell time is just counting how many consecutive time points the same state remains dominant or active. And then we can also use, it's another tool that exists from dynamical systems theory, which is to compute the transition probability matrix, which is also called the Markov chain. And basically, so when we have, for example, 5 states, what is the probability of being in state 1 and remaining in state 1? So here in the graph representation, the state 1 is this global mode that here is represented as transparent, but the idea is that all the signals are going in phase together. And then with 8% probability, it switches to another pattern that overlaps with the default mode network, okay? And then with 50% it remains there, with 20% it decays back to state 1, or with lower probabilities it can switch to other networks. Now, one thing that can be learned from this type of analysis, for example, is that we see that there's always higher probability to remain in the state than to switch, which means that they are at least weakly attractive in the sense that they maintain the activity there, but they never get stuck there as well. So they eventually fall down from them. But always, whatever state you are, if you don't remain in it, the highest probability is to decay back to state 1. So this is the first column in the matrix that you see is lighter blue, and the other transitions are less probable. So there's always here, we also represented this with thick arrows. To decay back to state 1 is always the highest probability. So this suggests that this baseline global state is what is interpreted as a main attractor, because then it's never always there, but then the others can be approached as pseudo attractors, or a more fancy name or provocative is ghost attractors, in the sense that these networks are consistently detected across subjects, but they don't last much. So we see them, like you saw in the video, they appear and disappear, right? So they are recurrent, So across subjects, we consistently detect the same pattern, even if for a very short period of time. So it's interesting because this reveals that what fMRI, it's like an approach to approach what we are capturing with fMRI from this perspective of dynamical systems theory. And then how does this relate with brain function? So now that we have this metric and we can compare across conditions, and the first time I applied this tool, or when I was developing it actually, was for a collaboration where we had fMRI scans from 88 healthy older adults. They are all more than 50 years old, and they were classified as good and poor performers. So this was from a cohort of 1,000 people, and they did some clustering analysis, PCA, to detect, categorize them according to cognitive performance. And among the best and among the worst, some were called in for fMRI scan. And the typical analysis were not showing many differences, and we applied this technique, and it revealed that apparently the good performers spend more time in this global state. Like this was the most significant difference, was that they have higher probability or higher occupancy and higher dwell time in the global state. And the poor performers had more probability of exhibiting the visual and the somatomotor network. So this was published at the time. It was the first time we're getting some type of results, but still, like, it was important, like, to understand better what this is. But anyway, it revealed that there is actually a change in this exploratory dynamics, let's say, of the brain at rest. Then we applied this to other data sets. For example, in a data set from schizophrenia, it's an open source data set from over 100 participants. And we found that people with schizophrenia had, like the poor performers, reduced occurrence of the global mode. And here we really did the overlap with known resting state networks. So they also had less of the default mode network and less of this orbitofrontal network. And they had more occupancy and more excursions, like we see here, to visual networks, somatomotor, and limbic networks. So there are alterations, like statistically significant alterations. Here we really increased the statistical power. So this Miguel was a student from applied maths who really helped in making the statistics wrong. So these results are really there. Even if we still don't fully understand what is going on, there is an alteration in these dynamics of functional networks in tests, in line with what has been reported in the literature. But this is just to show that applying even this algorithm also shows this relevance of these patterns. For example, we also found that it relates with, there were significant differences between controls and patients who had at least one episode of major depressive disorder, but they were not, they were in remission. So they were not depressed at the time, but we wanted to see if there were already some signatures or alterations. And actually it revealed a network that for the people in psychiatry makes sense, because it involves the salience network, but together with the subcortical areas, including the dorsal striatum. So this network was significantly decreased in controls. And not only it would occur less in controls, but when it occurred in the controls, there was more probabilities to switch from this network to the prefrontal striatum, to a network that connects the prefrontal and the striatum. Whereas in patients, they had a different type of pattern that was more the prefrontal limbic. So the prefrontal is more about reasoning, right? And how to where the prefrontal is connected seems to be associated with its function. So with controls, they link the frontal with the parietal areas, and the patients link the frontal with limbic areas. And they had more probability then to switch back to the global and visit less this prefrontal striatum. So it's, again, interesting, Then I will show here some examples of different studies and data sets that I applied this to. So here we applied this to patients with obsessive compulsive disorder, and the strongest difference this time was in a network that involves subcortical regions. So here, if we see the list of brain areas on the right, the areas. So this is like this is the eigenvector. The eigenvector is a set of values, right, that can have positive and negative elements. So I represent it here as a bar plot. But we see the areas that are in red, that have an opposite sign, which means that they will evolve in anti-phase or will be anti-correlated with the others, are exactly the basal ganglia. So it's the caudate, the putamen, the pallidum, and the thalamus in connectivity with the anterior cingulate. And this connectivity between basal ganglia and anterior cingulate has long been hypothesized and related in the literature to be related with obsessive behavior. So there is some interesting things that this methodology or these patterns relate to. And then, for example, we found that these networks, again, relating the orbitofrontal areas here, this one significantly increased with music listening. So when these are early adolescents that were listening to music or silence, and then also a dummy piece. But here, the difference between no music and music was highly, was significantly different, with a p-value of 10 to minus 6. And it involved this, again, these areas, these prefrontal areas that are associated with reward processing. And also it correlated with the musical reward sensitivity. So it's, again, strange how even just music alters the patterns that we observe. Then with deep brain stimulation, we found there were actually 3 different networks that showed different. One of them was the motor, of course, but we found that the people with deep brain stimulation also had significantly more occurrence of this pattern, here a bit represented differently, but these bars are with deep brain stimulation off, so inside this, so the people can turn on and off the deep brain stimulator, right? There are, of course, some movement artifacts, but here we compare with controls from this first LEiDA paper, so these are healthy elders, and we find that the occupancy of this network when DBS is off shows no significance, no difference with the controls, but when DBS is on, it's highly different. And then this is another paper we did it was in collaboration with a group from Oxford, which is called The Power of Smiling, and we actually found which. So the method was to see which network correlates the most with the happiness score of the pictures of the babies that the people would be seeing inside the scanner, and appeared a network that again involved this orbitofrontal cortex, but also the insula. And the thing was that the babies were presented with neutral face, but they knew if they were happier or not. But so it related with memory as well. So it was a bit with learned infant temperament. And actually the network that came out, this was the only one that was significant, related the orbitofrontal cortex with the insula, the amygdala, and the hippocampus. So it relates with the memory and emotion. So it's interesting to see this sensitivity to capture these patterns and how these patterns relate with brain function. Then also these patterns can be modulated, as I mentioned earlier, by drugs, or like I showed before with deep brain stimulation. But with drug here, it was a collaboration with the Center for Psychedelics Research at the Imperial College. And the paradigm was that the people were scanned, the participants, and they were first scanned without the drug. And then while they were inside the scanner, they would be infused with a compound, which is psilocybin, which is the active ingredient in magic mushrooms. So the people were really having a trip inside the scanner, or I don't know. And also 2 weeks apart, they were scanned again and given a placebo. And they never knew, like sometimes the placebo was in the first session, sometimes the psilocybin was in the second session. So there's no subjective effect here, but of course then they feel if they are with us. And the interesting thing here, why I find it really interesting, is that we first compare only the signals before and after psilocybin, right? And it's the first two bars. And we found that this state 3, the executive control system, so laterofrontoparietal, the one associated with cognitive function from the Lancet paper, significantly decreased with a p-value of 10 to minus 6, I think here, with when in the session after DMT. And then we took the placebo session, which was not yet pre-processed in the conditions. We take the centroids that we, like these centroids here, we run the clustering, but only one iteration to match to these centroids as well. And we find that in the placebo session, there was no significant difference with respect to the before psilocybin, but significant difference with respect to the other. And also, you see here the stability of the occurrence of the other networks across sessions. So the probability of being in this limbic network is always between 10 and 12%. Of being in the DMN also didn't change much. And the only one, so when this one decreased, the frontoparietal, what we observed was more of the baseline, like the hypothesis before I showed that it was like this main attractor. So in a way, one of the ghosts or the pseudo attractors seems to be inhibited by psilocybin, and this doesn't affect the occurrence of the other networks. I found this particularly interesting, and also this was recently replicated by another group that I never knew. I just saw the paper coming out. So they also used LEiDA on another data set with psilocybin, and they also found that it affected the frontoparietal network. So it really corroborated the results. So it shows again that the statistics are not lying, even if they're not super strong, there's something there that, that is replicable and consistent. So overall, so far, this is just to summarize a bit. So we applied this to the data set of the Human Connectome project of 100 healthy adults. And we found that there's a range of occurrence of these different networks. So they don't really, no one showed 50% of the time the DMN or others, but there's always very consistent values even across people. The values were quite reliable. Even if they were consistent across people, it was possible to identify the person itself by the probability. So people who had more global mode in one scan had also more global mode in the second scan. And people who had less global mode also had less global mode in the other scan. So there is some reliability in that. We use this interclass correlation. Then in terms of altered values, because this is also giving us insights on the biomarking potential, right? So we found in terms of psychopathology, let's say, in major depressive disorder, in schizophrenia, in autism. And now I'm applying to the large data set, ABID, with 1,000 subjects, and we are seeing that the people with autism have less of the default mode network. With cognitive decline, auditory hallucinations, either clinical or non-clinical, obsessive compulsive disorder, like I showed. In terms of even preclinical symptoms, for example, we found that there was a relationship between the occurrence of the default mode network and the capacity of people for trait self-reflectiveness, because it's something that people with psychiatric disorders have less ability to, I don't know, to look at themselves, right, and to analyze themselves. Also, alterations after people suffer from a relationship breakup. So it also correlated with how depressed, how sad they were, because they are not clinically depressed. But it's interesting because it goes beyond just the clinic. So maybe some preclinical values. And then another aspect that is useful when we do this type of analysis is to understand what tunes these values, right? Like I said before, so we have this effect of psychoactive drugs with psilocybin, and now with DMT, finding again that the strongest effect is the suppression of the frontoparietal network is the first thing, and now I have p-values 10 to minus 8, so it's getting stronger. Deep brain stimulation appears to change this. Well, here I put computational models because we're using this to tune this probability of occurrence of these different states in some computational studies. And then these natural stimuli that also seem to affect. So if seeing positive things or hearing music that we enjoy can help stabilize some networks, it can also be used if someone has, I don't know, as a therapy. There is an interest for, because I'm collaborating with the Center for Music in the Brain, and there is some interest on that. But then, so that was to summarize that I'm fairly convinced that they are related somehow to brain function. So it's not just an artifact, right? It is there, and because we could be detecting them all the time, but it was just an artifact. But or a physiological thing. But even if it's physiological, because I believe it may even be linked with physiological processes, it relates with brain function, and that is the message. And now it's a second part, let's say, of our third part of the talk, is about what are these networks, right? And some people call them networks, others call them gradients. More recently, now they're called waves. They are detected, so that we can call them modes, let's say, to be consistent with the different nomenclatures, but they are detected by distinct analysis methods, right? People use either principal component analysis, independent component analysis, co-activation patterns, k-means clustering with different things, because instead of clustering the leading eigenvectors, you can cluster the sliding window correlation. There are also method called quasi-periodic patterns, hidden Markov models, dynamic mode decompositions. But overall, they all converge to this repertoire of modes. So it's like discrete modes, right? It's not like a continuum, that are defined in space and with weights varying in time. Okay. But overall, like what is their nature and what are their organizing principles, I think is what the field is really needs to go into that direction. Because, for example, here I'm putting a paper from Nature Neuroscience from this year, I guess. And again, they are saying that they can decompose into 3 modes. And then here in this plot, what they show is how much the 3 complex principal components that they detected overlap with the CAPs, the HMMs, so different types of networks detected across different modalities. So I think we have reached a consensus that these networks are there, and depending on the method, they may be slightly different, but now we need really to understand where they come from, right? And so, as I showed before in the first slide, we have these signals, and the current hypothesis is that these are like aperiodic co-activations of the hemodynamic response function. And then, as I put here some arrows, so since these areas are connected together, this is assumed to be, so the fact that these areas co-activate at the same time says something is propagating across the structural network, like the question. But then there is this, but then if we look really at the signals, whenever the signal increases in those brain areas, it simultaneously decreases in others. Like the DMN is not just characterized by, so the rest stays at 0 and the DMN activates. It's like the nodes in the DMN activate, but the others go down. You see, like the signal goes down. There are points in space that have no motion, but there seems to be this counterbalanced activation. So this is what in the eigenvectors give the positive and negative signs. And this simultaneous co-activations and deactivations, they point to what is observed in the phenomena of resonant modes, let's say standing waves. Okay, so this aligns with, I will now go in that direction. But what's important to capture is that these are fundamentally distinct mechanistic scenarios that both could give rise to the positive patterns of the network of the DMN. But the hypothesis of the propagation across the structural network doesn't explain why there are some areas that lock in antiphase, right? Why when some areas increase, the others decrease?
Because there's no long-range inhibition at this scale, so it's
following mechanisms. So this concept of standing waves in brain dynamics is not new. Here I put the paper from 2012, but the hypothesis of standing waves is much older, from the 70s and 80s, and Haken and Girsa have been proposing this to explain EEG and showing these spherical harmonics. Then, for example, in Selena Tassoy in 2016, she used the connectome, so to show that you can also get waves or resonant modes in networks, right? So she applied the graph Laplacian. So the Laplacian is the eigenvectors of the Laplacian is the way to get the resonant modes of a system. And she applied the graph Laplacian to the connectome structure and detected some modes, again, that they call the connectome harmonics, that in a way proposed that what we are seeing are resonant modes in the connectome. And then more recently, the group from indeed Alex Fornito and Michael Breakspear, and led by, well, first author was James Pang, they showed that even if instead of considering the graph, the connectome Laplacian, the graph Laplacian, if you just use the surface mesh and apply the Laplace-Beltrami operator, which is another way, but to compute the mode, to estimate the modes or predict based on the surface alone. And they also found a repertoire of modes. They applied it only to one hemisphere, so they show it from the side, but like here with like the first mode here was the left right, and I think they were still missing the global mode. Usually it's discarded in the Laplacian. But still there was some alignment, so there is here a competition whether these are modes of the network or modes of the surface. But overall, irrespective of if it's from the network topology or the surface geometry that is giving these modes, overall universally standing waves or resonant modes are associated with the principle of resonance. No, I'm sorry, because this image is sad because it's not. Let's see what is going on. Yeah, it's coming back. Okay, so what happens in a mode of resonance? So what does this mean? So any system, be it like just a string, or here I show it as in a membrane, it can have multiple modes of resonance. So here in the first one is the fundamental mode where everything is in phase. You see the one on the left. Then the second mode, which is like half of the system goes up when the other half goes down. But what's important is all the constituents or all the points in this system organize, or they lock in phase when this mode is active. And the same system can have multiple modes of resonance, right? So this is something that is observed across fields and scales. And while here in the video, as illustrated here, they are oscillating forever, these standing waves, like, they are not always in a limit cycle, like I explained before. They're not always like self-sustained forever. Usually they are damped. So they oscillate for a period in time and then decay, right? Or they can be over-damped if they decay very quickly to a fixed point. So you just activate the mode and then it vanishes down to a static point. So the thing is, could the macro scale intrinsic networks that we detect with fMRI be standing waves? Sorry, so here I'm repeating a bit what I was explaining earlier, that we can calculate this, make analytic predictions from the structure, either based on the connectome or either surface-based. Overall, they always exhibit this gradient-like phase distribution, similar to functional networks, but the overlap of these modes is only approximate. So even in the paper in Nature, they just compared with doing the graph Laplacian, but we still don't know if that's the ground truth, right? They are still just approximations, because the modes of resonance depend not only on the surface, but also on the resistivity of the medium through which the waves propagate, they depend on the boundary conditions as well, so the elasticity, so there are different properties. But maybe, yeah, again, to do an animation to convey what is the concept of a standing wave. So I hope this is getting a bit slow, but let's see if it stabilizes. Anyway, of waves, the nomenclature is slightly different from the network theory, because in waves, the nodes of a standing wave are points that do not move in time, okay? So like I'm pointing here in the bottom. And the antinodes of a standing wave, they do oscillate in time. They are the points that have more energy, right? Usually when we see there's an illustration with plates, they are called Chladni plates, and you put sand on it, and the sand accumulates in the nodes. So the sand goes to the points of no motion, right? But the parts we don't see without sand are the parts that would be oscillating faster. But what is relevant here is also different antinodes. So the antinodes organize into two, they have two polarities, let's say. As you can see, whenever some antinodes go up, the others go down. So this can drive functional connectivity, let's say, or correlated signals between 2 points that are distant, but just through the propagation across space, so the resonance of the particles, right? The particles are not necessarily moving from one place to the other, but they are hitting their neighbor, hitting their neighbor, hitting their neighbor, so it's propagating across space, like sound waves, like standing waves. So then the question here is, we saw that these networks that we saw that could be ghost attractors, like what is the attraction, right? What is the principle that could explain why these networks are more stable than other types of configurations? And one explanation could be that they are linked with resonance phenomena indeed. So they would be, whenever you enter in a resonant mode, since there is this constructive interference or resonance, right? This network would be, this pattern would become more stable than others, and you could be switching, you could be playing overall these different modes of activity. Of course, this would be oscillating. So to understand actually this resonance phenomena, trying to disintegrate this, it's important to look at the fMRI signals. We have to see if they oscillate, because the standing wave will oscillate in time, right? But with the double resolution we have in fMRI, it's hard to see if there's sustained periodicity between some patterns, or at least if they have some angular momentum. I mean, this means that even if they just oscillate for half a cycle or a quarter of a cycle. And so I met Noam Chamesh from in Portugal, who has a preclinical scanner for MRI. And I asked him how fast we could go in fMRI, even if it was just a single point. I needed to see if the signals just fluctuate or if they oscillate, because that would be a way to see if it's related with resonance at some level. So we did ultra fast acquisitions and from a single slice of the rat brain, and we reached a TR of 38 milliseconds. So we did a 10-minute scan, and in each scan we had 16,000 frames from a single slice. So we don't change the slices. Usually, even in humans, what takes long and makes the sound is the slice changing. So with this, we found indeed that the intrinsic modes in rats could resonate like standing waves. Here I need to wait for this to stabilize a bit. It's very important that we capture. I mean, this is after 38 seconds, and the video. My computer is getting a bit slow. I'll just try to do here to see if I have another presentation open, or if something else is. I have one here. I'll close this one. I'm sorry. I'll just close this one. Okay, let me see if this time it gets better. Okay, now it's better. Okay, so the idea is really to show that I will now explain better how we reached that. So we did.
Okay, maybe I deactivate the video and then reactivate to see if it will be better. Yes, we have some trouble with.
Or maybe I deactivate my own video.
Okay, I'm deactivating the video, just a few seconds.
Okay. the Zoom. You can also stop my own video. Sorry. Let's see. Are you back?
Yes.
Yeah, I think now it's working.
Okay.
Now it's working. I think I put my computer to the limit. Anyway, here for these experiments, we had to turn off the alarms of the scanner, and it was really, we were controlling the temperature. It was really pushing also the MRI scanner to the limit. But we did, just to show the paradigm, the rats, we did 6 rats only, but the results were quite consistent and we had a lot of time points. So they were first just sedated with medetomidine, which is a drug that is known to kind of enhance the detection of resting state networks, but also to show like strong oscillations. Then we added light anesthesia, just with a bit of isoflurane, and then we added deep anesthesia. And in each condition, they were scanned twice for 10 minutes. And we also had 2 postmortem scans. So here first, we analyzed the resting state activity from the rats in different frequency bands. So what I'm showing here, for example, it's, so imagine we have a scan from a sedated rat, and we filter the scan in different frequency bands. And then I'm plotting here how much power there is in each voxel, in each frequency band. So for example, and then this is averaged across the 12 scans in each condition. So we find that in the sedation condition, there were strong oscillations detected up to 0.3 hertz, but peaking here in power around 0.2 hertz. For the rat brain, this superficial layer is the cortex, and here this huge part that is in blue is the striatum. So we see some activation of the striatum at slower frequency bands. So this extends beyond the typical resting state range used even in rats, because people in rats use the same range as in humans, but indeed we find that this extends beyond. So we also detect it in the typical resting state range. And then, as we give a bit of anesthesia, the power decreases mainly in these cortical oscillations. And when we increase the anesthesia, only power at the first frequency bin, so below 0.05 hertz, so it remains. And here, this is the postmortem scan. So this is always power relative to postmortem. I'm showing here at the bottom the mean and standard error of the mean power across in each of the conditions across all voxels. So we see that, for example, in blue is the power in each frequency band. In blue, no, in green, in the sedation condition, right? And you see here that it's high up to 0.25 hertz and then falls. With a bit of anesthesia, the main difference, so here it's the same as with the sedation, and then here it decreases mainly in these higher frequency bands. So that's why people give a bit of anesthesia to the medetomidine sedated rats to mimic resting state activity. And then with deep anesthesia, you see here, this is so in the first frequency band, they all have high power, the 3 conditions, much more with respect to the dead animal. And at higher frequencies, the difference with the postmortem was not significant. And above this, because this is also, we put this in the supplementary, but also deserves some explanation. Above these frequencies, we did detect strong power only at the breathing frequency and the heartbeat frequency. And so our Nyquist frequency was 12 hertz, which means that we could in theory detect oscillations up to 12 hertz. And I thought we could even maybe detect oscillations that link with EEG, but no, nothing that was significantly above postmortem was detected. So, but this is just to see. Okay, so in this range, which is where we found the most significant differences between the 3 groups, okay? So you see the rats sedated with medetomidine, light anesthesia, and deep anesthesia. I will now show a video of the activity in this range. Because, for example, here we could have power in this frequency band, but we don't know if all the voxels are oscillating in phase or if they are how they are organized, right, in time. So this is the signals I now will have to wait a bit because this is again at high resolution, but it is okay. So these are the signals, the scans from 3 different rats, all in the same condition. Please play again. All in the same condition, and we see similar patterns of activity, right? So this is not driven by a single rat. This is what I'm trying to illustrate here, that whatever is happening in one rat, we see similar waves happening. Sometimes they seem to be going from the bottom to the top, from the top to the bottom. It's unclear like in which direction they are going, but there are some waves. If we add a bit of anesthesia, the activation seemed to be more global across the cortex, like whenever it turns red, it's red inside the whole cortex or blue, but we still see signatures of the same. And with deep anesthesia, put this from the beginning again, and hope that it will play. Okay. Anyway, in deep anesthesia, we adjust the scale, of course, of the color map. I do like this, maybe it will show, but we can see that it's almost noisy. I can tell you that, okay, only, for example, here in the middle rat, we see it's a bit more red. So there's this global activation, but apart from that, apart from this global positive and negative activation, sometimes it turns more blue. It's very similar to what we were detecting in the postmortem animals. So postmortem animals, you see something like this. It's just noise, right? There's no hidden thing. So we applied principal component analysis, which is the most simple way to detect modes of resonance from empirical data. So to detect the eigenmodes of the system. So basically we do the covariance matrix and then get the eigenvectors. And when we get these eigenvectors, as I said, the eigenvector, the sign of the eigenvector doesn't matter. So you can have it represented positively or negatively. And what we find is when we take this eigenvector and then look what is the temporal signature of this eigenvector. So let's say this is a n times n matrix. It has 1,400 voxels, let's say. So 1,400 is 66. The eigenvectors are 1 times n, right? And the signals recorded are n times t, the size. So when we multiply the eigenvector back to the original series, we can get a 1 times t, right? Because we contract the n dimension. So we get a temporal signature that we can then compute the power spectrum of that temporal signature. And what is really nice is that we find, for example, here, there are some modes that really have very sustained periodicity. So they really oscillate in time very strongly, which would not happen if we just average the global signal here. It's only when you apply this kind of mask that you detect these oscillations. So these principal components have nodes and antinodes, right? Like you see here in space. So it's interesting because we really see that there are points of no motion, which would be the ones in green. And then there are points, the antinodes would be the dark red and blue, and they would be in antiphase. And again, what is nice is that this can drive long-range functional connectivity. Even if, like according to this principle, you wouldn't even need an axon to connect these two areas for them to be correlated together, right? This could be happening in a tube or in any volume. So it's interesting because then if we, let me see now, I hope this will work, this video here. But here is if these are eigenvectors, okay, they are orthogonal, right? So we could in theory just sum them, right? So imagine on the left is the recorded signals. I will play the video now, I'm just waiting it to charge a bit. And then here are each of the principal components, so the spatial modes that are obtained at the group level. That's why they were so nice, because they're really clean, because it's like the average of all the eigenvectors, like I do the average of all the covariance matrices and then get the eigenvectors of that. and but multiplied by the temporal signature from a given scan, of course, from this scan S that is shown here. And then we sum them all together. We sum just these 10 wave patterns and show, okay, let me see, please, should play well. It's annoying because this is like one of the most important. Okay, now it is going. Let's see, it will reconstruct. Anyway, the nice thing is that the activity that is reconstructed seems, okay, not only to denoise, this is something, of course, that PCA does, is to denoise, but what I want to show is that the activity from a single scan can be reconstructed from the superposition of waves detected from even from other animals, right? And we can explain, so this ghost patterns that we can see in the normal fMRI signal could be explained as the superposition of standing waves. And what is most important, because people say, are they standing waves or are they traveling waves, is that when the superposition of standing waves drives traveling waves, drives the illusion of a traveling wave, right? So we could see traveling waves in different directions and observe complex patterns of dynamical functional connectivity, because if you then just try to look at the patterns instantaneously, there's an infinite possibility of patterns just with the superposition of 10 modes, right? So it's interesting to show that maybe, like, I don't know, like what we were showing before on the connectivity between the networks, then maybe the network and the connectome is more for the EEG activity and network interactions, but maybe what we're seeing in fMRI may be more related with resonance phenomena. Now, when we add 1% isoflurane, not only we found that there was a lower number of modes, okay, again, the number of eigenvectors that we detect is compared with, so we take only the eigenvectors that have an eigenvalue larger than the largest one in the postmortem scans. So in the postmortem scans, we do the same. We take the leading eigenvector and look at the eigenvalue, and we say, okay, anything at this level or below is noise. We just want to see the ones that have more eigenvalue or more energy, let's say, than what is detected in the dead animals. So here, I mean, the bottom ones appear a bit noisy, but here at least these 4 ones, they are slightly different, except for mode 2. But importantly, you see the power spectrum of these modes is more broad, so they are less fine-tuned. We see some oscillations, there's some sustained periodicity, but it changes between frequencies. So here it may oscillate at slower frequency, then it goes at faster frequencies. So there is this strange effect. And then when we have isoflurane at 3%, only one mode passed the threshold, and it didn't have sustained periodicity. Okay, and it's like aperiodic fluctuations. We saw like slow fluctuations. So we see in the carpet plots like everything blue and red, and that was it. So how to analyze and to compute statistics between these things? So what we did was to take the modes, so the temporal signatures across the 20, so the 12 scans in each condition, say, so each scanner scanned twice. And then we calculated both the peak frequencies, I'm not showing here, it's panel A, but also the resonance Q factor. And what we found is that under medetomidine, so the resonance Q factor is how many, imagine how many cycles the wave pattern will oscillate before it decays down to 0. With medetomidine only, for example, for that animal I was showing, there were some animals for mode 2 that they would resonate like almost like 12 times. So we would see it going up and down several times before it decays down to 0. With a bit of isoflurane, this resonance Q factor decreased, and with medetomidine, it seems like they either become overdamped or what it is. And here I plot the autocorrelation function of the temporal signatures. So remember, we had these temporal signatures here. We can do the autocorrelation function to see, like with the phase lag. So we see that if we do this, imagine here is for 200 seconds in terms of lag, but we find that with medetomidine, for example, this is for mode 1, a certain rat. It's mode 1, same rat, except for in different conditions. Here, it does 3 cycles before it decays below a given threshold. With a bit of isoflurane, it does just 2 cycles, and we see that it's clearly slower. And with isoflurane 3%, it really, I mean, it passes here the threshold below, but then it's almost similar to postmortem. You see here how we represent. And then we try to represent this in the face portrait, also to make the link with these damped oscillations. But in this case, these damped oscillations are not for single nodes. Now the damped oscillations are for wave patterns, right? So the thing is that, okay, they are over damped. Here is also just to show a video. Here I'm really filtering in the narrow frequency bands of this rat had strong power at 0.2 hertz, but it varies a lot the peak frequency between animals. But this is to show that, so the mask is not done manually. I'm just putting, showing the voxels that have power in this frequency, and then showing their temporal signatures. So you clearly see here, there seem to be a traveling wave going from the bottom to the top, but one hypothesis is that these traveling waves are propagations, like pressure waves in a way. So there's not necessarily a net flow. So there's not a particle that is here that went up. So when it's just a propagation of some form. And then we also think, I don't know, but in these cases, the damping is proportional to the resistivity of the medium through which the waves propagate. So it's possible that isoflurane anesthesia alters this property. We also did the modes at the whole brain level, also like just to see how they would look like. So they are pretty nice. And then the idea was also to try to make a model, like we are saying, since we want to try to understand what are these models. So, okay, previously people have just tried to predict these modes from the structure. Okay, I assume these modes. I accept. Let's say okay, these are standing waves. Maybe if I had the good model, I could predict it from the structure. But here, what I want to see is what if these waves behave as underdamped oscillators, right? So this underdamped is the damped, let's say. So as waves that when they are perturbed with noise, imagine in the resting state, these modes could resonate. It's like echoes in the system. If you just apply noise, maybe you could hear or feel these modes. So we take the modes defined from the data, we simulate time series with the same frequency band, just matching the peak frequency and the damping coefficient. Okay, so that's what I tried. So here I'm just showing 100 seconds. Before I was showing 600 seconds, but this really approximates what we were seeing. So here this should have a similar resonance Q factor and the same peak frequency as in the scan. And we simulate, and then the simulated brain activity in this case is the spatial patterns, each one multiplied by a given oscillator, noisy oscillator. And then we show that we observe, like here is the different snapshots of the activity simulated. And if we do the correlation matrix of this, then of course we would obtain a similar patterns as observed in typical functional connectivity analysis, which is when they put the seed in one place, they find the activation in contralateral voxels. And so in a way, this replicates these patterns of long-range functional connectivity without assuming any network connectivity. So in a way, I think understanding the origin of brain modes may shed new light in the missing link between brain activity and brain function, right? Like, what is this translation? What are these macro scale patterns? So I think these results support or align with the hypothesis that brain modes represent standing waves intrinsic to the brain structure. So relating with a paper by Alex Fornito and James Pang, and actually in OHBM, we were talking in the same symposium. Now I'm already booked to go to Australia in February as well, because I mean, this is a different way to support the same hypothesis, right? To look at the temporal signatures, but then there's still a lot to do to understand how these brain modes are orchestrated together, like what is their relationship with neuronal activity, what is the relationship with neurotransmitters, with physiological processes, and how does this translate into mental function? Like, do these gradients, let's say, because they form dipoles, so it's a bipole. So in a way, the fact that these areas, for example, in the orbitofrontal cortex, so if there is a mode of resonance in those antinodes, there will be more energy, right? Or they can have also some attraction force to things that are, I don't know, some type of poiesis, some types of motion to attract neurotransmitters. And for example, to attract the increase of glutamate in those areas. That is what I was going to say. Like, does this have a role in driving the organization and less a consequential role? So again, I thank all my collaborators and thank you for listening. Okay.
So thank you very much, Joana, for this fantastic presentation. And let's move to the audience for questions. So
thank you for your presentation. Could you back to the slide number 12?
Yes.
Thank you.
Sorry. Okay. Now, yes.
Yes. Maybe this is not the correct one, but I just want to know that how has it been shown that there is a kind of attractor in the system?
Okay. Yeah. I mean, the fact that I don't know, there may be over 100 or 200 studies detecting the same pattern. So imagine if you had a dynamical system, each one had a water balloon, and everybody's analyzing different water balloons, and everybody detects the same pattern in the water balloon. I mean, it's an attractor of the system, which means that there is it's a state of a system where the system tends to go to. And what I feel is that there is so much literature consistently detecting very similar patterns. That was the point of it, in a sense. Maybe I'm over-expressing my ideas, but in a way, I think there is sufficient evidence to at least make the hypothesis that that these patterns that we observe in brain activity that are consistent and reliable may be linked to some attractor dynamics. I mean, these are also very conceptual let's say aspects, but I don't even know if there's a way to prove it is an attractor. But for example, some ways, this is what I wanted to do, the collaboration with Bruno Cessac, who is a professor in dynamical systems, and he gave me all the theories. I was saying, well, you partition your state space and then you calculate the probabilities, and if the diagonal, so if there's always more probability to remain than to switch, for example, you can say that. But then the thing also with the concept of attractor is, for example, in multi-stability, a system that has multiple attractors, it would need noise to switch from one attractor to the other, right? So imagine you have these different wells. I don't know how far, but from what I learned, at least in quantum mechanics, like there are these different duels of attraction, and then you can either switch from each state or each attractor by noise, or if the attractors are naturally unstable or metastable, so they remain there, but then they decay naturally, they would be pseudo attractors, so they are unstable. But the thing is that we don't know if this is driven by noise or not, if it's switching. This is an approach. Then what I'm showing is the superposition of standing waves goes a bit against this, because here we would be switching from one state to the other, to the other, to the other. And according to the standing wave hypothesis, it's more like they are always they are there present. It's just their relative amplitude that changes. And so actually what we're seeing here is which of those waves is dominant at each instant of time. And we look at the leading eigenvector. So yeah, but there is no, I don't know. There was a paper from Victor and Girsa and Gustavo Deco and Victor Girsa proposing this multi-stability and ghost attractors. And that's why here with Gustavo and Jakub Wolicek, who is also like from maths and physics. So they wanted to, so the idea was to formalize a bit these concepts, but yeah.
Thank you for your explanation. I just want to add a point. Maybe it would be better to say that what you detect is a kind of equilibrium state. And we can say that this kind of equilibrium state can be an attractor. I mean, this is a kind of conjecture based on...
Yeah, you are right. You are right. Indeed, equilibrium state seems like a very nice word. And indeed, this would be different points of equilibrium. And why the equilibrium? Why these points are equilibrium points? Perhaps because they are modes of resonance, right?
Okay, thank you. Any other question from the audience? No, I have few. So just referring to what it has been done in the literature with several people looking at the consistency of the different network, and this is following what it has been said right now about equilibrium point, or so the what is well known now is that few networks are more consistent than others. So all the networks who are like in the sensory-motor system are very consistent over object, but few more the cognitive ones are less consistent. And there is a nice paper published by recently by Bratislav Misik at McGill, and who show that. But previously,
so because it's across people, so you are saying different people have slightly
absolutely.
So it difficulties the detection,
Exactly. And there are other papers published by people like Yves Bourneau and his team in France, where they show, they introduce the notion of this consistency. What they show is the consistency is over what they call the ring. And this, based on the resting state networks, they develop the theory of dual interwind ring architecture. So it's mostly what they show is that all the sensory motor systems are within one ring, and the other system are within another ring, and they are communicating between them. So this is a very interesting point of view, and this is also related to the biology, because they make some correlation with the genomics data, and they show that the characteristics of the genome support this kind of this two-ring architecture. So just my question is about consistency. Well, I think that what you replicate is probably very strong in the sensory motor system. So what about the more cognitive networks?
Cognitive, like executive, well,
Executive, salient, and all those networks.
yeah, the executive control. I mean, I was showing here for the sorry, for the psilocybin, but I have the plot also here in voxel space for this. But it was very, it's really the lateral frontoparietal. If I put the list of brain areas and even the, so there are lateral frontal, I mean, areas. I don't know. I mean, we also, like, regarding this variability across subjects that you mentioned. Indeed, then when I go for each single subject and look at the mean of all the occurrences in the individual subjects, it's hard to, we don't, like, they don't always overlap very well, these different networks. But here the thing is that if we don't take a common basis, it's very, very hard to compare the occurrence of these networks across conditions. Right, because we want to see. So here maybe the approach, yeah, maybe I haven't evolved in other networks. Of course, I mean, here I'm not showing the pyramid of centroids. I usually cluster from 2, 3, 4, 5, 6, 7 up to 20 states, for example, and see for which clustering partition do we detect better these differences. For example, in this paper here, it was for 7, for k equals 7. It was when it better detected. So it's how it better detects this network. And maybe indeed it needs to be a coarse parcellation because of so much variability across subjects. I mean, a coarse in the sense that if I start subdividing even more the networks, I can see other patterns appearing, but then it's hard to make sure that they will appear in all the scans. You see what I mean in everyone. So
Yes.
maybe, yeah, we could decompose this. Yeah, it would be interesting to see, to make a better study. I always try to have a common basis, you know, a common repertoire also in the, so to be able to compare. But yeah, indeed, maybe it would be nice to see how much they vary. But I mean, it's nice. I already saw, I saw a talk from Bratislav this year, and I know, and I'm happy to see that people are going in, yeah, in other directions. For me, at this level, is really to understand where, what is the and especially what is the origin of the signal and the contribution of interstitial fluid in this. But it will relate to what you were saying about the glutamate and all these things, because there's contributions that we are neglecting in the signals, in the fMRI signals. The fMRI is very sensitive to fluid dynamics. And even if we remove the signal in the ventricles and in the subarachnoidal space where CSF is, the CSF enters inside the brain as well. And when you're awake, the CSF, the interstitial fluid, is locked inside. When you're asleep, it flushes, but when you're awake, it's closed there. So there could be some resonance phenomena happening at that level. I mean, it's hypothesis. I need to test these things, so I cannot make any, it's just speculation.
Okay, so this is very interesting. If you come to this recurrence of activity patterns, I'm just trying to make a link with what it has been published also in the literature, who support also what you mentioned here. We got, for instance, the work of Melanie Bolly. She is a Wisconsin University, or Guillaume Marilec, and they work on this functional integration and segregation between networks. So they define the networks, and for each individuals, they look at the relationship between the networks from the informational theory. So what are the exchange between how much information is exchanged between networks and within networks, which is related to this
the superposition. That's what I was thinking, because in a way I've seen that some people try to see how much, for example, you can have at the same time the DMN and the frontoparietal activated, right?
Exactly.
Or like, is there a functional relevance for having the global simultaneously active with the DMN. I mean, here the second mode is the default mode network, or for having it simultaneous with another. And indeed, this will translate into different patterns. So it can, when you have different standing, like you can even make things move in one direction or in the other, just depending on which one comes first and which one comes after, you know, in terms of network activations. So there could be some functional role of this interaction between networks that could drive the transfer of information from one area to the next, or even at the collective level, because I'm not sure then I think things then are integrated at the collective level. We may be able to sense everything, right? So not just, I mean, it's not about transferring it to somewhere and we will feel when the information is transferred in one place, but yeah, we feel from everywhere. So it's,
yeah, so what we expect is that all the networks are involved. Maybe we have some network who drive the activity at some point, because if we come back to the principle of the decomposition that you are using, a PCA technique, it's so we got orthogonal component. So if we suppose that the underlying noise in the data or the data are Gaussian, this means that we are extracting independent component. So we don't expect having independent component. We expect having more related components. So it's very interesting to see what does it mean, each component, regarding the dynamic of this component.
But if they are modes of resonance, if you perturb a system, a resonance system with noise, I mean, of course, it eventually decays to the fundamental mode. It's the last one. But if you perturb with noise, you may detect multiple. Each wave or each harmonic is orthogonal, I would say, to the previous. And they don't necessarily interact. I mean, it's like, for example, imagine, sometimes I take the analogy with music, in the sense that the different chords don't interact, but then they create music. You know, like at the emergent level, at the higher level, music is created from the superposition of nodes that don't know what the other is doing, but for some reason their superposition creates a new thing, right? You use some different nodes, and if they are out of order, you don't create music, and if they are in order, you create music. But music is not something that then can be extracted directly from each. So I don't know. Yeah, there are these two different components in the perspectives, and I mean, if you compare my first talk in the morning with this one, because I think one of the questions was aligned with that, I'm like, these seem to be two completely different things. And what I see is that there are so many perspectives to look at the same thing, that it's important really to keep our eyes open to different hypotheses. And like you said, like I will see the others, and I like to debate with other people, because I think if we need different hypotheses and different things, and then to prune them, right? To do experiments and tests to validate whether one or the other makes sense. But yeah, this one I think aligns more with this resonance hypothesis, and resonance don't interact, waves don't interact together. That's why we hear things, that's why we see things, that's why because waves don't interact, they just superpose.
Yeah, I agree. So it's a very interesting point of view. My last question is about
coupled oscillators, and I was using the coupling function to couple the oscillators, and these are not coupled. So there is this strange, Sorry.
My last question concerned the isoflurane and sedation. So I'm referring to some work we did a few years ago with the team of Pierre Maquet from the Université de Liège, and with isoflurane in healthy subjects. And what we observe is this decrease with the concentration of isoflurane of the connectivity between area, and we apply, I mean, tools like functional integration and segregation between also networks, and we observe this the decrease of this the relationship between the networks, even the structures of the networks, is disaggregated somehow.
Yeah,
So,
I totally found this as well. Yeah, there's no, I don't detect any mode, only this global mode. But actually, this global mode extends beyond the brain alone. It really goes to all the tissue.
So at that time, we conclude that the isoflurane has a huge effect on the bald signal, which makes sense. My question is,
But you don't think it acts also on the neuronal signal?
probably yes, but I think this is more related to what we debate this morning about the hemodynamics, probably the metabolism, and we talk about astrocyte, and I think the huge effect is at the level of the astrocyte, who link the capillaries to the neurons. So I'm just wondering, what do you think about this kind of effect regarding the result that you have?
And I mean, I know that, so isoflurane cannot be just affecting the contrast, it's affecting brain function, right? So in a way, whatever we are seeing in the signal is also at least correlated with the loss of cognitive functions, because in deep anesthesia, it's, I mean, it's one of the definitions of consciousness is what you lose with anesthesia, right? And indeed, we lose many things. So even the signal indeed just looks super noisy, like I showed with deep anesthesia. I was also showing at a higher frequency. If you filter, so it's actually at 0.02. So it was like one oscillation every 50 seconds. So we see here, you see here, it's one oscillation every 50 seconds. So it's very, very slow. What I was detecting in isoflurane, that remains the only thing with some power that is still there. Compared to the postmortem, because in humans we can never do what is, because I wanted to see what is because the animal is still alive, right? So we should be able to detect something different compared to the dead animals. But yeah, indeed, it's not just about the bold signal being lower, because then we would still detect the other networks, but at lower power. It's really about the different organization of the signals, like I mean, they're not organized anymore. There are no equilibrium points anymore. There's no resonance. That's what I think. I mean, I don't know, but whatever we are seeing here. And that was a bit the hypothesis, that it destroys the properties of the medium through which these waves propagate, or whatever it is, or like you're saying, if it's the glia cells that are in charge of propagating the fluids, because they are known to have these aquaporin channels, right, and everything. Maybe, yeah, indeed, they allow or not the fluids to pass and to give this signal. I don't know.
Yeah, I am very curious to apply your approach on a data set that we had several years ago, but the data set come from patients or called CADASIL patients, and they have a neurodegenerative disease, but the disease is at the level of the capillaries only. And the neurons are not concerned by the disease. So what we observe with the bald signal, of course, the bald is decreased because there is like a disease at the level of the capillaries. So I am curious to look at this kind of things, because there is like a segregation between what is going on at the level of the EEG signal in those patients where EEG seems like quasi normal. But of course, the bald signal is completely different and maybe and close to what we observe sometimes in Alzheimer's disease. So it would be very interesting to see how this your approach can detect the patterns, and how those patterns can be consistent within if there is a signature of the disease within those patterns from the dynamic point of view.
Yeah, that would be really nice. We are now also analyzing, we downloaded the whole ADNI, the Alzheimer's neuroimaging data database, and we applied the LEiDA, and indeed we find highly significant differences between healthy controls and Alzheimer's disease. And one of the patterns, the most significant patterns, is where the cortex is in antiphase with an area in the cerebellum, but that is very close to the fourth ventricle. So I think because there are now some papers showing that the signal in the ventricles are like this anticorrelation decreases with cognitive decline. So I don't know, but I think there is something very interesting to explore about how much the signals in the CSF influence the signals in the cortex, and because indeed that's what we're finding in the Alzheimer's data set, is this ventricle cortex, this pattern of antiphase disappears. It's there in healthy controls, and so yeah, it's strange, but maybe it just tells that people should be like looking in a more open view of not just focus, because in the resting state we are not just doing task related activation, right? So we are not really triggering the hemodynamic response function. So so it's important to understand what exactly is driving these oscillations and how they are propagating. But I mean, I have a set of experiments that I want to do. I ordered CSF, I have like a thing, and I'm doing some experiments, but it's hard to make conclusions. But, yeah.
Just a comment, a general comment, because several groups, and maybe the more famous one is the first group of logothetis, who make a link between EEG and fMRI, and using, for instance, the envelope of the EEG signal convolved with the hemodynamic response, and he got some kind of fluctuation of the Bolt signal that all makes sense, I would say. Very interesting, old result, I would say right now. And the question is, okay, if this model is correct, so we can apply the same approach as you did, rather than considering the Bolt signal, considering the envelope of the EEG signal. So in this case,
actually,
what is
this morning, I'm working on that exactly, looking at the EEG signature of the functional networks, because I have simultaneous EEG fMRI.
Okay.
So from what I can tell you, whenever the DMN, for example, is active, we see increase in alpha in the middle of the brain, right? So it seems that the alpha increases in the areas that are not activated in the DMN.
Okay.
And when the executive control network is also an alpha wave in those time points, but with a different topography, it has more alpha also in the frontal electrodes when the executive control network is active, like in fMRI. So we can do simultaneous, that's what I'm saying. We can see what is the EEG topography when the fMRI network is appearing. And then the global is more associated with delta waves, and the limbic more to theta, but I'm still trying to understand why these things are happening. But I also think it's very, very important to go in that direction and try to understand the link between the formation. But it's not that an increase in bold will reflect with an increase in EEG simultaneously. It's in different parts.
Yeah, probably,
I think
yes.
more idle rhythms.
Okay, so I'm curious to see the result. So, okay.
Yeah, because for example, when you open your eyes, you decrease your occipital alpha and you increase your fMRI signal in the visual cortex, right?
Yes.
So there is this negative relationship between alpha and bold. Yeah, so it's important to understand.
Yeah, that's what we see a few years ago with my student working on the how the patterns, what are the common patterns between the envelope and the Bolt signal. So for the EEG fMRI simultaneous acquisition, so Sokel and who is a PhD at that time, he did this kind of work, and we see some discrepancy between what we observe in EEG and what we observe, of course, in fMRI. But there are all the network that you mentioned, the default mode and the motor network, visual network, all of them seems like completely common to the two modalities, which is...
it's important to understand better this link. I mean, there's a lot of research, but there's still no, ground through like consensus, like this very convincing evidence of something of the strong link.
Exactly. Okay, so is there any last question from the audience?
You see here, it's night already.
Oh, yes, it's okay. Last question.
Okay. The sun is
yes. Thank you for your talk. Maybe a quick question. Maybe I missed something, so I'm sorry about that. But so at the end, if I got it well, at the end of your talk, you said that what you see primarily are standing waves that oscillates, and that this can easily be taken for traveling waves.
That's the thing. So
how can you be sure that it's not the other way around, that they are that they are in fact traveling waves that you easily mistake for standing waves? This I didn't get.
So the thing usually they do to illustrate a bit this, I mean, if you have a structure and you put points in random motion going in all directions, right? So imagine traveling waves in all directions. Eventually, the traveling waves that find an equilibrium, so that find a loop, let's say, that find a way to go in one, so to hit a boundary and come back and hit another boundary, they will resonate. And those will be the standing waves. Those are the intrinsic ones. Indeed, so standing waves are the result of the superposition of traveling waves going in all directions. Then so only the ones that survive will be the standing waves. So
that's some kind of inference patterns. Interference patterns.
Interference pattern, exactly. It's constructive interference. So standing waves result from constructive interference, but that is what the physics. In theory, we have in our minds that something is really traveling and propagating, like there is a displacement of particles really going and hitting the wall and coming back. But this is like pressure waves and things. So it's just like a vibration. So it hits the neighbor and the neighbor and the neighbor and the neighbor. So these waves can propagate across space, even if there is no net displacement of the particles or whatever is there, right? They can just propagate. And then they generate these standing waves. So this could explain why we detect them across subjects. They would be imagine they go, and then from time to time when they interfere, it creates like this wave, but then it's very short-lived because there are a lot of interactions and things, so you don't see it anymore. Then about what we see then at each instant of time, then it's this superposition of these standing waves. I mean, that's how I could explain why they link to functional networks. First, we have a fixed repertoire.
Yeah.
That's also another thing, is that intrinsic modes are discrete, like quantum. It's not like a continuous that you go from one mode. No, you have one mode, then you have another, and then you have another, and this could explain why people detect this repertoire of modes. But yeah, I mean, there's no certainties in this field at this time, but that's also what makes it fun, right? So we have a job for the next 50 years.
Thank you very much. Thanks.
Okay, so thank you very much, Joana. It was really a pleasure having you with us today and having this great discussion with you and the great lecture and this very nice and exciting research program that you have.